This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 101, recorded on March 26th, 2015. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello, everybody.
How are things on the West Coast?
Well, they're fine. We have a heat wave now. It's gonna be tomorrow's supposed to be 90 degrees.
Great.
Crazy.
You know, we still have a little bit of snow on the ground here.
Sure.
Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello.
The world traveler.
Yeah, I had the pleasure of being invited by the Swiss Society for Infectious Diseases to participate in their 37th annual symposium.
Where was that?
Lucerne.
Lucerne.
Not bad.
It's nice. Do you like traveling, I hope?
I do. I'm also teaching, so it's a little tricky to shoehorn it in, but you get time on the plane where nobody talks to you, and it's fabulous. You get a lot done.
Yeah, unless you have a gregarious neighbor.
Right.
I bet you could just shut them up, though.
I give him that look.
Yes. Also joining us today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
How have you been?
I've been well.
Chamber of Commerce weather, right?
Today, the sun has finally come out. We've had a rainy week, but I'm hoping that we'll stay in spring and not move to what Elio's seeing in San Diego is full-blown summer.
Oh, that's amazing, 90 degrees.
Yeah.
We're just above freezing here, and there are lots, if you pass by the parking lots of shopping centers, there's still these huge piles of black snow.
Yuck.
Ooh. It's very ugly. You must have that in Michigan, right?
The last of my snow is gone, I'm happy to say.
You didn't get any on Monday?
Nope. So we're cycling, but not yet golfing.
A few weeks, right?
Soon.
So we, last episode, of course, was number 100. And so today is 101. We're like teaching an introductory course today. Today is
I'm sorry I wasn't there because I was going to institute a round of For He's a Jolly Good Fellow directed at you, Vincent.
That was
off the ground.
I'm
but
glad
our listeners are going to be spared our singing.
Okay.
Well, that's probably good, at least for me.
In the past week, however, we passed 1 million downloads.
Wow.
1
That's
million.
a lot.
All the episodes, 100 episodes. That's about 10,000 per episode. So we have lots of fans out there, and we appreciate them and thank them for listening.
Great. I'm using them in my undergraduate course as a teaching tool, and it's been really fun.
What do you do? Play him? You don't play him in class, do you?
No. I'm trying to flip the classroom. So I choose TWiM or TWiV that discusses a topic of broad interest. Michael, you'll be pleased to know we talked about copper this week.
Oh, cool.
So they listen out of class, and then we go through the paper kind of.
Great.
In small groups, they'll discuss a figure and then present it to the class.
There is a way to ensure that they actually listen, right?
Well, the podcast really helps them understand because these are freshmen, and so they don't have the expertise to understand the written paper, but the combination works.
Well, that's our intent.
So my hat's off to you all.
Well, you're part of it, so it is unusual. You can't get recorded conversations among scientists, and I think for students that's really great to hear. To hear us talking and discovering, and
saying I don't know.
Yeah,
and catching us in our mistakes. When we do make mistakes, we like to get letters, and we like to acknowledge that we've made a mistake. And so, if you're out there listening, help us out.
Yeah, if you hear something wrong, let us know. TWiM@twiv.tv. Our sponsor for this episode of TWiM is ASM Press, and they'd like you to know about a new textbook called Principles of Microbial Diversity, written by Professor James W. Brown, who's been teaching microbial diversity for over 20 years at North Carolina State University. The book sells for $90. You can find it at asmscience.org. Today we have a snippet and a paper for your listening pleasure, and I'll hand it over to Elio for a snippet.
Okay, so let me start by proposing that you have some friends over for dinner, and you open up a bottle of wine from California, and you pour a little bit into a glass, and you twirl it under your nose, and you say, these are the words of an old New Yorker cartoon, you say, well, it's a simple domestic, but you'll be amused by its pretensions.
Yes, in New York.
And then you drink it a little bit, and you detect flavors like apple, pears, bananas, a flowery aroma with a hint of honey, and your friends are very impressed by your savvy. But what you've really told them is that you smelled ethyl acetate, isoamyl acetate, and phenylethyl acetate, plus a whole bunch of other stuff, okay? And this is all due to our friendly yeast Saccharomyces. And it turns out that this is not widely appreciated by people outside the business, that Saccharomyces is a most obliging organism. It does two things. One is alcoholic fermentation, which is what you think of first. And secondly, it makes a whole slew of volatile compounds which add to the aroma of the wine. And a good wine is not just selected by a special grape from a special terroir, but also by the kind of yeast you use, because different yeasts are going to give you different proportions of these compounds. So that is the story that I'm going to tell you about.
I'm thirsty. You made me thirsty, Elio.
Okay, well, you can go out and have a bottle of Chablis anytime you want.
And he's at an Ivy League school, so it's a wet campus probably.
Yeah, I'm sure.
Unlike our state schools that are all dry.
I'm sorry. I'm very sorry.
Yeah.
Michigan is dry?
It's dry.
Oh, my gosh.
Well, suffer in silence, please.
Yeah.
Anyhow, so what's in it for the yeast is the question. Why is yeast selected to do this? And by the way, it turns out that the laboratory strains of Saccharomyces don't do much of that. So if you want to make some home brew, don't use the strain you have in the lab. It won't work very well. Go out and buy a strain from you can buy the special strains for special purposes. Obviously, yeast and beer are different strains and so forth. Anyhow, so what is it? So there's a paper, the title of which tells a story. It says, the fungal aroma genes ATF1 promotes dispersal of yeast cells through insect vectors. Before I tell you what it's about, it's by a bunch of Belgians by the name of Christians, Franco, Koels, De Meester, Michiels, Wenzelaers, Hassan, Yaxi, and Westerpren. And these are people who, some of them, the senior author Westerpren has written a lot about the subject of yeast products. Anyhow, the story is very simple. It's told in the title. Some of these volatile compounds, which we so appreciate, which give the flavor to wine, are smelled by fruit flies. Fruit flies love yeast. In fact, they eat mainly yeast. In the laboratory, when you want to grow fruit flies, you grow them on yeast agar. So they smell this stuff and eat the yeast, but they don't eat all the yeast. Some of the yeast end up on their
hairy legs.
The legs. What's that?
They're hairy legs.
The hairy legs. That's right. They have hairy legs. There's a lovely picture, if you look at this paper, which shows you a nice fluorescent stained spots, two spots on the leg of a hairy leg of a fruit fly, and that's due to the yeast, which has been modified to contain fluorescent proteins. So the yeast eat the fruit flies eat the yeast, but as I say, they don't eat them all. And anyhow, yeast can also make spores, and these spores are fairly resistant. They probably can go through the intestinal tract of the yeast unscathed. Not a bad story, right? So everybody benefits. The yeast benefits, the Drosophila benefits, we benefit. Couldn't be better, right?
Mm-hmm. So you mean the good
there's a but coming.
The good smell of the wine isn't just for us?
That's right. No, there isn't actually. I tell you what, there is not a real but coming. The story is that good. So how did they study it? Let's have a look at what this paper is about, because it's a fun paper. So they characterize the strains, and they find out that a mutation in this gene ATF1 abolishes the formation of certain of the higher alcohols. Not all. There's a big table here of lots, about a couple of dozen compounds, and it's the acetates which are involved in this production, okay? And so in the mutant, you don't make this. By the way, this is a diploid strain, so they had to go to a lot of trouble to delete both copies of the gene. That's not so easy, but they did it. Anyhow, the strains which have this gene active and the ones that don't have the same growth rate. So this is not required for growth, but it's required for formation of these volatile compounds. So it turns out that now they have a wild type and a mutant, the mutant being in the production of certain volatile compounds, acetates, and they test that for attraction to Drosophila. So they build a little gadget with 4 corners, and they put they make the air that goes in can be modified. And so they have, in one of the 4 corners, they have a control, 3 are controls, and one is the experimental where they add these compounds. And when they do, the flies are attracted to that particular corner. And they automate this whole thing. They have a visual automation gadget so they can measure how many flies go there. So this is neat, and there's a very clear distinction. The preference is really very big. And it's to these acetates, phenylethyl, isoamyl, and ethyl. Okay. And the most important of this is ethyl acetate, which is, I think, the smell of pears.
Mm-hmm.
I'm sure that Michael knew all this.
No. I think the thing that made the paper come alive to me was their magic little chamber with the vacuum in the middle, and it sort of looks like a 4 square, and then they show you a picture of where the fruit flies are, and that below that picture is the graph showing you the fraction of the flies. And if you go to the supplemental, there's wonderful movies showing this. I mean, the flies just eventually all end up in one corner, piled up one on top of the other. Oh, yeah.
It's pretty neat. You can watch the flies tracking across the juicy yeast colonies, and it's quite visual.
That's right. It's really very visual. So that's a great story right there. Okay, so they're using the trick is using a mutant. The mutant is specific for these activities, and so it really follows up very nicely. Nice work. But then they do a little bit further, and they measure the neuronal response of the Drosophila. And that's complicated stuff, and I don't want to get into it because we don't have to, but let's just say that they have a way of measuring the activity of the neurons in the anterior lobe of a Drosophila brain. And sure enough, the responses in the wild type are clear. There's a response to these esters, and there is no response in the material from the mutant. So the yeast odor is caught in the antennal lobe, and the yeast behave accordingly because their nervous system tells them that. And that's almost it, except for one more experiment, which I thought was really quite neat. And it shows that this gene, ATF1, which is responsible for making these acetates, also impacts on the attractiveness of the yeast cell towards the fly. And the way they do this experiment, and so this results in increased spread. And you can say the whole point of the exercise for the yeast is to be spread to different environments. Okay, so the way they do this is they, again, in this case, they have yeast which are labeled with fluorescent proteins, and they put them on a petri dish, essentially, They put the wild type and the mutant on the same part of the petri dish, and they have a fly running around, and they show that there is many more of the wild type than of the mutant spreading or being carried around by the fruit fly. So, since for yeast dispersal is essential, this is really a neat experiment. As I say, the title tells you that this gene promotes ATF1 promotes dispersal of yeast to the Drosophila partners. That's it.
So
nice
elegant. Really elegant.
Simple and nice, and it gets you into thinking about good things like fine wines.
So if you made wine with yeast lacking ATF1, probably wouldn't be very good, right?
It would be a rot gut.
Rot gut.
I think I drank some of that once.
It would not be worth drinking.
I have to say, I love red wines, but I can't smell anything. I don't smell pears or whatever else you said there. I just like it. I don't smell any of this. I don't. Do you smell pears? And hint.
I actually smell the alcohols. I smell the alcohols because I think I did too much organic chemistry, so I never smelled the fruit. And so I associate it with isoamyl alcohol and some of these other complex alcohols that we used in molecular biology to extract DNA.
I smell the berries and cherries and
that you can smell.
That family.
If I read the label, I can smell it.
Yeah.
If I don't, honestly, I don't know what they're talking about. A hint of this and a hint of that. But there's a language. And the people, I mean, I have a friend who's a vintner, and he tells me that, there's a lot of hype in what they put on the label, but there is some truth in it. And that people who are trained to do it actually can tell. They can tell the difference. They can give names to it in terms of known aromas. But gosh, you have to be very good at it.
This whole issue of odorants for microbes is just gaining lots of momentum. We've done quite a few papers now here on TWiM and on TWiV about how, on TWiV in particular, how viruses induce certain organics, and those promote dispersal, just like this. It's quite interesting.
I saw a headline to a paper where malaria-infected cells make an odorant that attracts mosquitoes. Wow.
I saw that. I just saw that as well. Yeah, and that's very common. And we got into a discussion. Someone brought up a paper on TWiV where infection of animals can make their urine have odorants in them. And then the purpose of that we don't know. It may repel or attract other people, other animals to be infected. But then people started sending us letters about how you can tell if someone has cancer by the smell of their urine, and there are dogs that can smell this and so forth. So this has gone on, and it's very interesting. Odorants, very important stuff. Without them, the world would be boring.
That's very true.
Thank you, Elio. That's very nice. I love it. Yeah, fruit flies. I can just smell the yeast, they're so characteristic.
My graduate work was in a yeast lab, and I used to love coming into the lab in the morning and opening the incubator and getting that
whiff,
right? Smell of yeast. Yeah.
Yeah, that's right.
Does it remind you of the lab, or does it remind you of fresh bread or wine or what?
Well, it's a pretty intense odor. Would remind me of the lab first.
Okay.
You don't have any good odors with Legionella, right?
They're a little smelly, but it's a respiratory pathogen, so of course we don't put our nose down on the plate and smell.
Yeah, that would be bad.
Mm-hmm. Can I go back to the wine for a second?
Sure. Yeah.
Here's a line you can use anytime. You want to impress people, you do this shtick of pouring a little wine and smelling it, and you say, travels well.
Travels. What does travel mean?
I don't know. It means it comes from California or it comes from France,
I see.
You didn't shake up the sediment in the bottom to make it stinky.
No, right. But you have to pretend to know what you're talking about, right?
Yeah.
I went to a tasting once, and this guy's house, and he had hired some professional to do it, right? And one of the things the guy said, he's trying to teach us how to smell things, and one smell smells like a horse running through a barn.
Oh, God.
That's not pleasant.
No, that's not a good wine. When it runs through the barn, it's no good.
Can I close this segment with my favorite New Yorker wine cartoon?
Yeah, sure.
There's a couple who's hosting a dinner party. They open the door to greet their friends, and the couple hands them $30 and says, "Here's the money we would have spent if we would have stopped to buy a bottle of wine."
Nice. Yes. Really nice.
That's actually good, because that way you can buy what you like.
Nice. All right. Well, many scientists do like wines. I encounter them all the time. So thank you, yeast. Our second sponsor for this episode of TWiM is the ASM Education Department, and they'd like you to know about a special issue of the Journal of Microbiology and Biology Education, all about scientific ethics. It's guest edited by Dr. Beth Fisher, Dr. Frederick Grinnell, and Dr. Michael Zygmunt. It's a 45-article section that provides a comprehensive look at ethics in science, and includes the responsible conduct of research, ethics in science education, scientific publishing and authorship, and ways to incorporate ethics into STEM education. And these articles provide ethics-related activities and tools that you can use in a classroom or a laboratory setting. This section is also essential for the ongoing education of active researchers and research mentors. You have to keep up with your ethics. They provide articles on why responsible conduct of research training is needed in the research environment, ways to incorporate and evaluate responsible conduct of research training, and it discusses the potential influences that may compromise research integrity. To find this special section, go to jmbe.asm.org. Let's get on to our paper, which will be explained by Michael. We're continuing with Staph aureus. We did this for 100 as well, right, Michael?
Yes. And we're moving from the pleasant odor of wine to the pleasant odor of dirty socks. The paper is entitled Transmission and Microevolution of USA 300, MRSA or MRSA in the United States households, evidence from whole genome sequencing. And for those of you who are uninitiated, MRSA or MRSA stands for methicillin-resistant Staphylococcus aureus. And if you've never smelled Staph aureus, it literally smells like dirty gym socks. So since we're along the lines of smelling, and this paper is by Alam, Reed, Petit, Boyle-Vara, Miller, Els, Dom, and David, and they're from three institutions principally: Emory University, the Harbor UCLA Medical Center, and the University of Chicago. And this paper was published in the most recent March/April MBio. And when I read this, I couldn't imagine that we've already got 6 years of this journal under our belt. It seems like only yesterday that we were launching this journal at ASM's general meeting in San Diego. And so the papers in this journal continue to get better and better. And these authors present a very curious story about a microbe that our listening audience has likely heard about, and that's the infamous MRSA. And most of us have a general idea that staphylococci are normal constituents of our skin, where we basically have two principal types. We have the ones principally without coagulase, and that generally encompasses the species like epidermidis, hence the term Staphylococcus epidermidis. And then there's other things like the staph that live in the crown of our heads, and that's Staph capitis. And then there's a new emerging pathogen, londinensis, which was named for the city of Lyon, France. And those of you who are Latin scholars will appreciate that londinensis is actually Lyon in Latin. And this londinensis is the seemingly harmless skin commensal that can transform itself into a life-threatening pathogen, causing infective endocarditis, just like its more virulent cousin, Staph aureus, which we're principally going to be discussing here today. And all of these staphylococci have one thing in common, is they like to live on the semi-arid, somewhat acidic surface of our skin. And the intent of this paper is to try to understand how this very significant pathogen, USA 300, is able to invade our homes, because USA 300 is community-acquired MRSA, and how it invades our homes and then goes from being colonized on individuals within our homes to causing infections in people, and some of those infections can be pretty significant. So I thought I'd start off by giving you a little bit of a background. This particular strain of Staph aureus first arrived on the clinical scene about 15 years ago in the early 2000s, first amongst football players in Pennsylvania, and secondly amongst prison inmates of Missouri. And Fred Tenover and Richard Goering, Fred was formerly at the CDC, and Richard Goering is a European scientist of significance, and they wrote a review in Antimicrobial Agents and Chemotherapy to specifically introduce us to USA 300 in 2009. And in their abstract, they actually tell us what to look for. And the reason I'm bringing it up here is because it's old tech. And the subject of this paper, as its title implies, is they're doing whole genome sequencing. So in 2009, whole genome sequencing, while possible, wasn't very efficient and it was very expensive. So Dr. Tenover and Dr. Goering described USA 300 as a methicillin-resistant Staph aureus that was a particular pulse field type, and that's what USA 300 represents, is it's a pulse field pattern. And it was also then typed via multi-locus sequence typing, and it was clonal complex 8. And then they looked at a staphylococcal cassette chromosome marker, specifically the methicillin cassette, and it was type 4. And what they described is over the next...
What does all that mean? What does all that mean?
It's a specific technique, the specific techniques that the taxonomists have been using to categorize various strains of staphylococci.
So what's so special about this strain that it got so much attention?
This strain, it's actually predominates in the United States. It's associated with the community. It's not associated with hospitals, and it predominantly starts or enters our home as a cause of a skin and soft tissue infection. But this particular microbe, left to its own devices, can actually become an invasive disease. And it's this invasive disease that, of course, has caused all the hubbub, going from bacteremia to endocarditis to severe and acute necrotizing pneumonia, and also osteomyelitis. So it has this panoply of behaviors. It's going from
panoply, you mean?
Yes, panoply.
And it can also survive better than other strains of staph on the skin, right? It's picked up this locus that allows it to metabolize arginine and then neutralize the acidic pH on the skin. That's
the bit of contention that this paper reveals, and we'll get to that in the discussion. But Michele is spot on with that observation, because the Acme cassette, I always think of the Roadrunner and Coyote cartoons, where if you think about it, Acme was always supplying the dynamite that was trying to blow up the coyote. And that's effectively what this cassette is thought to help facilitate, allow this microbe to really neutralize spermidine, which our bodies secrete through our skin to actually inhibit the more virulent staphylococci from getting a foothold on us.
That's where arginine comes in, isn't it?
That's exactly where it's a part of the arginine operon. And it's this spermidine acetyltransferase that's responsible for neutralizing the spermidine. And it's part of this major Acme operon that's doing it. But before I get to the meat of the paper, I want to introduce this first digression, and I'd like to acquaint our listeners with a free monograph that was produced by the American Academy of Microbiology, where our very own Dr. Swanson is the chair of its board of governors. And in late 2013, they convened an expert panel and conducted a colloquia on the threat of MRSA. And this is a terrific and free report. You can download it from the web. It's written in lay language, or as my grandmother would say, it when something is completely inclusive, it goes everything from soup to nuts about MRSA. And Michele, would you like to comment on first the report and then the process of these frequently asked question colloquia that the Academy runs for the benefit?
Just briefly, I'll say that the American Academy of Microbiology has, as part of its mission, is to take advantage of its expertise to make key information that the general public needs to make decisions about their life available through these publications, which we call FAQs, or frequently asked questions. So they're terrific booklets. They're available to download from the World Wide Web, and I encourage you to explore their offerings.
And they're on a variety of topics about jobs and all sorts of interesting things. So
beer, cheese.
In fact, that's the most recent one, is about cheese.
Right.
So it's a really interesting thing.
Vaccination. So now there's another on vaccines.
Oh, yes, especially with the measles outbreak of late. Right. Maybe Vincent wants to mention that one on TWiM.
Yeah, we have. Yeah.
So now back to this really interesting paper that these authors have taken us through. So the question, we know that this microbe, USA 300, commonly causes skin and soft tissue infections, but occasionally it can actually penetrate and then can cause all sorts of bad things like necrotizing pneumonia, endocarditis, both life-threatening and often resulting in life-ending outcomes for the individual who succumbs to the infection. And this particular strain is the most prominent community-associated MRSA that we see in the United States. In Europe, there are other vehicles out there. MRSA is endemic on our planet, and there are different strains that have taken a foothold. So consequently, the authors embarked on this because whole genome sequencing is now less expensive and it's much more readily available to do. And in fact, at Emory University, their genetics or genome center is actually competing directly with the competition over the pond in England, where sequencing sort of started out of the Sanger Center. So the Emory group is really taking the lead on this. So the question is, why do whole genome sequencing? In this sense, we have all these other ways to categorize MRSA. And the answer is they principally wanted to be able to understand how to design decolonization strategies, and then understand how, when a strain is introduced into a household, if we could figure out how we might be able to get rid of it if it's potentially a bad actor, or if you think about where the microbiome is going, if we could figure out how we might introduce a opportunity to introduce a particular strain that will do good things and then have that strain take a foothold in the host. And so we're going to start out simply where we're going to understand one microbe and how it attaches to a household and how long it can stay in that household. And the data from this paper, principally in the form of whole genome sequencing, first and foremost established that households serve as the major sites of MRSA transmission, principally through this particular clone, USA 300. Secondly, the length of time and hence the adaptation time that the MRSA strain resides in the household results in what you would expect: simple point mutations and also acquisition of new knowledge via horizontal gene transfer and or phage, bringing in new information into the host. And in fact, that's thought how Acme actually entered the staphylococcal genome is through a phage introduction on one of these islands of pathogenicity that the phage actually carried forward when it got moved into it. And then through this whole genome sequencing activity, we'll learn whether or not the virulence decreases with time in these small human populations, if you will, or a household, addressing the common observation, or what is generally understood amongst microbiologists, is that when microbes are cultured under the same conditions, for lack of a better word, under lab conditions, they often adapt to their circumstances. And Michele will probably tell us how when you grow Legionella for a long period of time on a Petri plate, it sort of loses its teeth. It's no longer...
when you grow yeast.
When you grow yeast, exactly. It loses its ability to make good wine. Right. And so they're going to specifically teach us, or hopefully be able to address, whether whole genome sequencing will reveal whether or not this virulence goes away with time just by looking at the sequencing. And so this is an incredible amount of work that was conducted by this group. The methods were relatively straightforward. They did whole genome sequencing, principally by looking at 146 strains of USA 300 from Chicago, which is in the middle of the United States, and Los Angeles, which is on the West Coast. They both have very different climates. I mean, you've heard enough weather reports on TWiM to know our weather is different in those locations. And what they did is they had an operational definition. They started out with an individual that had a skin or soft tissue infection that was USA 300. And they obtained cultures obtained from these infections. And then they asked the simple question whether or not they could swab 3 locations of folks that they lived with in order to assess whether or not they were colonized. They started off with 350 index patients with skin and soft tissue infections, equally distributed between the University of Chicago Med Center and UCLA. The reason I'm telling you the numbers is not to bore you, but to illustrate logistically how challenging this is. Just imagine 350 strains. You got to put them on plates. You have to then isolate the DNA. It gets even squirrelier when you consider that the patients were visited 3 times before 21 days after treatment, which gave them the baseline, and then again at 3 and 6 months. So the number of samples just multiply each and every time. So they go through this process, and by the time they get ready to go, they had 1,162 people enrolled in this trial from their 350 index patients. And they found in their initial screen that Staph aureus colonization was at about 40%. And normally, Staph aureus colonization for football players in one study that was done in 2003 was about 5.3%. So you begin to see that they're seeing some differences. And USA 300 was the major player, accounting for 53% of the individuals that were colonized with Staph aureus. So the rule of thumb is the normal human population is colonized with Staph aureus, but very few of us, only about 5% or so of us are colonized with MRSA. And now that 53% of the household contacts are colonized with MRSA, after this one founder has been brought into your house, you can begin to see how this is of significance. So
I'm trying to put this together. Can you sort of put it together for me, would you?
All right. So what they did is they asked the question, can MRSA, specifically USA 300, persist for a long period of time after being introduced into a household with one individual? And it's sort of like a freeloading relative that shows up and that stays in your house and is reluctant to leave. And so how do you get them to go? And that's effectively their core question is they ask the question, I bring in MRSA to the house and does it persist? And that's what they learned is that MRSA not only entered the household, but it was literally transmitted to everyone else. And that 53% of the individuals in the household became colonized with this particular USA 300.
Do they know how fast this happens? Is this something that happens within days, weeks, years?
No, because the way they did it is they had their baseline values and then at 3 and 6 months. So
what's your guess, Michael?
It happens fast. I think it happens relatively quickly because we're shedding so many skin cells per hour, and we're picking it up.
So avoiding this transmission by quarantining the person with the bug, that wouldn't do any good, right? By the time you find out it's already spread.
You can. And this is where the football players bring the lesson to bear. We know from studies done with high school and college sports teams that if you share common towels, it's very important that those towels go through a bleach cycle to kill off the incident staph. If you just use soap, the MRSA can actually hang on to the cotton fiber of the towel and actually be transmitted. So you need to run it through a significant bleach cycle. And in fact, that's how it's been thought to be spread amongst sports teams. Last year, in the 2013-2014 football season, there was 2 National Football League teams that were going to forfeit a game because one particular franchise was bringing in MRSA with a few of its players, and they refused to let them use their locker rooms because they were so afraid of contaminating their teammates with MRSA. And it's, again, a skin and soft tissue injury. And football is, of course, a contact sport in which raw skin touches raw, bare skin to the other player. And so it's easily transmitted, which leads me to believe that this colonization happens very quickly because Staphylococci love human skin to attach to.
Very good.
So now we go back to their questions. And the first question is, can this USA 300 persist for long periods of time? And the way they approach this problem, and they use some fancy Bayesian skyline coalescent modeling to effectively do this. And I had been avoiding the Bayesian skyline coalescent modeling seminars. And I tried to put this together for us because math doesn't work well for me on the radio. And the near as I can tell, and please send me letters if you have a simple way to explain this, is they're really using a small genetic sample from a large background population. What do we know now? We know that USA 300 is the major community-acquired MRSA that's in the United States population. Whenever we see someone come into a hospital and we swab them and we genotype them, 9 times out of 10, they're USA 300. It's a rare event that there's something else. So what these authors did is they took a small genetic sample, and the coalescent modeling statistics is something that asks the question about ancestral relationships of samples taken from individuals that comprise a larger population. And what the statistics will enable them to do with this Bayesian modeling is take this coalescent, or the coalescent product will allow them to convert information into these ancestral genealogies about the population history and vice versa. In other words, did the chicken come before the egg, or did the egg come before the chicken? And that's what this Bayesian model will do. And it's principally looking at the genome, and they ran it through something called BEAST, which is this Bayesian statistical modeling. And they learned about the average mutation rate. And the way I always think of mutations, because bacteria can give us so many numbers, is their average mutation rate was 1.25 times 10 to the minus 6 mutations per site per year. And thinking about the genome size of Staph aureus, which is about 3 megabases, and its core genome is about 2.2 megabases. What this mutation rate tells me, it will take a long time to adapt Staph aureus to a special circumstance unless there was significant selection pressure. And their mutation rate was no higher or lower than what was previously published for USA 300. In fact, a study by Ullman reported a mutation rate of 1.22 times 10 to the minus 6. So to me, what that meant is that for every million cells, there will be on average 1 point mutation per site per year. So you need
What do you mean per year? I'm sorry, I didn't get that.
1
per
point
year,
mutation
did you say?
per site per year. So you need lots of mutations or lots of cells in order for a founder to come in. And, unless you're going to actually have a lot of horizontal gene transfer that's going to effectively do this punctuated equilibrium type of mutation where you get a big leap in phenotypic changes. So
for this particular study, knowing the mutation rate was useful because they could then look at the sequences and then deduce how long that strain must have been in the household.
Mm-hmm. Correct.
Right. So they were able to estimate that the household had USA 300 in the household for, on average, 2 to 8 years before the infection that required hospitalization occurred.
Correct.
Mm-hmm. And it's all based on this mutation rate and computational biology.
And if you really want to be troubled by it, if you look into the references of where a lot of the, and this is one of my clinical colleagues pointed out to me, is a lot of the references for this sort of type of work is not being published in Journal of Clinical Microbiology or Antimicrobial Agents and Chemotherapy. It's being published in, if you just look in their reference list, references 18 through 21, it's being published in things like Nature Review Genetics, Genome Biological Evolution, Genome Research. And so it really requires someone who's concerned about the clinical implications of this to really begin to understand what all of these computational biology type of studies really mean for a clinician.
And that's an opportunity, if I could jump in and just
Please.
Tell you a little bit about this team that published this work. So I was able to talk to Robert Pettit and Takir Alam, who both worked in Tim Reed's group in Emory. And the way this story unfolded is Robert Dahm and Michael David had published previously work out of the University of Chicago, where they had studied independently households in LA and households in Chicago, and just asked how persistent is MRSA in those households. When they presented their work at Emory, Tim Reed's group got excited about taking advantage of these large strain collections and sequence databases they had to begin to ask questions about what are the differences between the strains in LA and the strains in Chicago. And by doing computational analysis, bioinformatics, which was really led by Robert Pettit, who's a master's student who's now getting his PhD with Tim Reed, they were able to illuminate this beautiful Figure 3 show how within each household the clone is actually quite stable, but each household has its own individual MRSA stable isolate. So,
and literally the founder begets all the others in the house.
Right.
Somebody else in the house doesn't bring another MRSA in. Once it's in, that one stays put and prevents all other comers from getting in.
So it's this great collaboration between the group at Chicago and the group in Emory that brought the bioinformatic analysis. And I have to add just a few personal details. Michael, you'll be pleased to know that Robert Pettit's hometown is Charleston, South Carolina.
Really?
That's right. And Takir, who has a really strong track record in studying antibiotic resistance, first studying malaria when he was a PhD student in New Delhi, India, and then coming to work at the CDC for 3 years, looking at evolution of antibiotic resistance in Plasmodium, and then joining Tim Reed's group. So he had the expertise in mechanisms of microbial resistance to antibiotics, and then teamed up with Robert, who had the bioinformatic expertise. And so the two of them described just a great camaraderie that developed in the lab. They would share coffee and talk about their favorite national sports, cricket for Takir and football for Robert. And the product is this beautiful paper that you see.
You know, the rest of the paper is really a tour de force in discussing, you know, how microbes adapt to their particular niche. And so the next series of experiments that they did is they asked questions about this, what's happening to the pangenome. And, you know, simply put, the pangenome includes a core genome, which makes you...
You wanna tell us what pangenome refers to?
Yes, that's what I was just going to do. The pangenome includes a core genome containing the genes that are present in all strains. So all strains of Staphylococcus aureus, specifically then MRSA. And then there's a dispensable genome containing genes that are present in 2 or more strains. And if you think that MRSA is a strain of Staph aureus, and then there's finally a unique set of genes specific to single strains, and USA 300 is a single strain of, if you will, MRSA, or a substrain of MRSA. And the distinctions are not always biological, since they often depend partly on which strains are included in the analysis and which aren't. And so in the pangenome section, they asked whether or not there was this continuous reshaping of what we now know as USA 300. So again, what they did now is they took the 146 whole genome sequences that they had. And again, this is the value of Emory, why Emory is really the place that this had to be done. And then they took 49 other completely sequenced strains of Staph aureus, and then using another bioinformatic tool, the unsupervised hierarchical clustering algorithms, they then grouped all of the USA 300 strains into the same clade, which means a single common ancestor, and then they assigned a date to that. And that date they assigned was 1993, if I'm remembering right, Michele. And here they learned that the pangenome of the 12 strains from Chicago were missing this Acme cassette, which was postulated to contribute to the success of USA 300 as a clone. And so that's what I think was really interesting about the pangenome, is they were able to lose this Acme cassette, and that they very colorfully show in one of their figures, in which they have all the sequences in blue, and then the ones with the Acme cassette having been deleted are in green. And they, in defense of the Acme hypothesis, they said it was probably brought in on one of these pathogenicity elements, and they then further characterized this Acme element, and they found that it was a unique Acme element compared to others. So again, the power of this bioinformatic approach to ask the question how things are changing. And finally, the last piece of their paper is about this fluoroquinolone resistance, which was completely something else. It effectively said that it wasn't holding true to the same transmission patterns as the USA 300. Fluoroquinolone was likely brought in on another mobile genetic element that was more promiscuous than the unique traits that make it USA 300. And so it's likely that in America, as strains move, whether it's as a consequence of these contact sports where sports teams are moving amongst our country, or incarceration where inmates are moving amongst our prison system, once the strain gets into a house, it can stay in there 2 to 8 years. And then, as Michele already suggested, before that strain actually is either triggered by a host response or the strain somehow adapts to the host to cause a fulminant infection.
Or someone in the household has an open skin wound, and that gives the microbe an opportunity
to penetrate deeply.
Yeah.
Well, the moral is clear. We all need to live alone. We can't have any sports, no sports, and we just have to drink red wine.
Well, the one thing I didn't introduce into this, and this is what Richard Goering, who's from I want to say the Netherlands, but it may be Denmark. And Rich, don't send me a note. I'm just being a little spacey this afternoon. And he looks at pets and how pets can contribute to this. And as we know, our pets can often influence staphylococci movement, and whether or not you're a pig farmer as well can influence whether or not you're moving MRSA about.
Michael, last time we talked about how a strain of Staph aureus went from people into rabbits. Number, right?
Yes.
That was, I think, ST121.
Yes.
So what's the relationship between that and USA 300?
They're just a little bit different by pulse field type and what they can do.
Is the 121, is that methicillin resistant as well?
That I don't remember off the top of my head.
Because I'd hate to have the rabbits, get that.
Michael, can I come back to this? The question about whether acme is accounts for the ability to survive on the skin. I think there's pretty clear data that indicates it can confer that, but that doesn't mean it's the only way that MRSA can. So there are lots of ways to skin a cat, right?
And I was going to point out that there's another paper in MBio that the emergence of epidemic methicillin-resistant Staph aureus strain USA 300 coincides with horizontal transfer of the arginine catabolite mobile element and SpG-mediated adaptations for survival on skin. And was the first author on that paper was Paul J. Planet, and it was a group out of Vincent's own Columbia University.
That's right.
And that was published in November/December in Volume 4 of MBio. So Michele's absolutely right on that. And so, as she said, there's more than one way to skin a cat.
Or a rabbit, perhaps.
Or a rabbit, perhaps.
That's right.
Or a human.
Well, we know there's definitely many ways to skin a human with this particular microbe. And that's one of the cool things in that monograph. It shows you all of the so many virulence factors associated with Staph aureus.
Nice.
And we can link to this
we will.
monograph in the show notes for anybody who'd like an easy access.
We sure will. Absolutely. It's very good. All right. Thank you very much, Michael. Lovely. And thank you, Michele, for pointing out that paper that was your find. I guess you're interested in Staph aureus.
Yeah, I happen to be teaching it right now. So.
All right. There you go.
So you've read on it.
Our final sponsor for this episode of TWiM is ASM Microbe 2016, a brand new meeting, an unparalleled event that brings together ASM's general meeting and ICAC together under one roof. It's going to be the largest gathering of microbiologists from across the planet and the premier event that showcases the best microbial sciences in the world, the only meeting in the field that explores the complete spectrum of microbiology and provides a true transdisciplinary experience. I have to tell you, I've been to both meetings, and putting them together is going to be phenomenal. Microbe will offer all of the high-quality program of the general meeting and ICAC, as well as targeted transdisciplinary sessions, a special profession of microbiology track, an expanded poster hall, topic-based networking opportunities, track-specific lounges, opportunities for continuing education, and much more. There'll be nearly 200 sessions and more than 5,000 posters across 7 program tracks. The meeting provides a unique forum for interaction among the global community of multidisciplinary scientists. And it brings together leading product and service providers to give you the best solutions to tackle your most pressing challenges. The call for sessions for ASM Microbe 2016 will open on April 7th and continue through May 19th, 2015. It's a single call that will inform the work of the General Meeting, ICAC, and the Profession of Microbiology Program Committees. These are the ones who develop the program for the meetings. ASM members can submit session ideas for the 7 tracks. You can go to asm.org/microbe2016. Let me read a few emails for us. The first one is from Eric, who writes, Dear TWiMers, I am a retired computer programmer and also a free market libertarian.
Uh-oh.
I listened to a recent podcast where the subject of mandatory hand washing by food workers was tossed around. I would like to present a few more of the free market arguments on this subject. 1, with a government mandate to wash hands, what makes you so certain that this mandate will actually have the effect that food workers obey this rule, especially if there's nobody watching in the restroom, and that they will do so effectively? And
None.
And he goes on to talk about washing your hands and so forth. 2, if there were no government requirements for washing or inspections, don't you think there'd be a niche for some certification agency like a consumer's guide to take the job of inspecting facilities? We all use electrical devices, and these come with ratings, UL certifications. If such a sign were displayed fraudulently, then even in a free market culture, this would be a criminal action. Free markets still have rules against force and fraud known as the non-aggression axiom. 3, when the government regulates an industry, this tends to make people complacent and to not do due diligence. We see this quite evident in the financial markets. In addition, the industries being regulated tend to get regulations that keep out competition more than anything else. And where do you think the regulators come from, if not the industry itself? Much incentive for fraud here. 4, if you and your friends keep getting sick after eating at a particular restaurant, don't you think the word will spread and thus weed out the bad establishments? And 5, surely doctors and hospital workers must be quite aware of the issue of cleanliness, yet in my own personal experience, I almost never see a doctor wash his hands before or after an examination. What is so special about food services? Why is there no sign at the doctor's office requiring washing of hands? I don't know about you, Eric, but my physicians all wash their hands.
Mine always do too, before and after.
Yes.
And all the healthcare workers.
Yeah.
And I just went to the dentist this morning, and even my dentist washed his hands before he put on his gloves.
What do you mean even?
Glove up.
Especially.
Yes.
There are many more points I could make, and these have been made by more articulate writers. One of the best is Murray Rothbard. I would suggest some of his works. I find it almost universal that anyone in the fields of research tend to assume that there is simply no alternative to the government when it comes to funding. I'm sure you all have numerous cases of waste that arises from this form of funding. In a free market, universities would self-fund their research and then perhaps create documentaries to pay for the research. I'm sure you have all seen how political favoritism influences who gets the grants. The reason I'm still not convinced by the climate change arguments is that I challenge you to find some climate change skeptic being funded by the government. I see no reason to believe that food workers are being regulated any better than any other line of work. However, I still love the podcast, although my favorite is TWiP. I like the new format with case studies. Keep up the great service. I don't know. Any thoughts, Michael? You like to talk about washing hands?
I talk about with things at work, and hand hygiene we know works. And
should it be mandatory for restaurant workers?
Well,
I don't see why not.
I see the reason. The sign is what's mandatory, and you hope that they follow the behavior.
Yeah, you can't enforce it, as he says. Right?
Yeah, you can't enforce it. And if we didn't have the rule, there would be no sign.
I mean, if surgeons didn't scrub, you'd get infections and you die, right, more often.
But there they have a very strict code of ethics, and they also have malpractice. And if they don't behave properly, their malpractice insurance goes up. And it goes to his argument that the free market will self-regulate. But at the same time, there's ethical considerations in that. And I guess we have to introduce ethics to food workers so that they know not to make people sick. And, how to do that for somebody who's getting paid a very, very low wage without, while still being able to turn out the food as quickly as they need to turn it out.
Yeah.
To combine all regulations in one basket seems to me to be a little bit not nuanced enough. There are regulations that are regulations. I mean, some that deal with finances have a totally different character than those that deal with hand washing. I'm not sure that it pays to say all regulations are bad because of a philosophical stance. I mean, it's just not true. There are some regulations that work perfectly well, and there are some that work less well. So you can't say because it doesn't work in Wall Street, it won't work in the toilet of a restaurant. It's apples and oranges, it seems to me.
I also think that if you put a sign up and you require your employees, but you say you don't enforce it, probably a certain percentage are going to do a good job, and maybe that makes a big impact, right?
And it can set up a culture, habits.
Yeah,
that's right.
Habits.
I also equate it to stoplights. I mean, we all know that there's a consequence of running a red light while driving. You'll get a ticket if there happens to be a police officer. But if the general public wasn't following the tradition of stopping at red and proceeding on green, we'd have chaos on our roads.
Yeah.
And that's effectively what happens when we don't wash our hands, is we truly do have chaos in the food system.
All right, the next one is from Steve. I wonder if your team has anything to say about the prevalence of salmonella in seed foods. For some years, I've been getting regular notifications from the English Food Standards Agency when seeds and seed products have been withdrawn due to the presence of salmonella. What made it all the more puzzling was that despite the huge furor there had been over salmonella in eggs, the UK media seemed not the slightest bit interested that so many other foods were still being regularly withdrawn due to salmonella contamination. I did ask the FSA to explain this attitude some time ago, but it didn't really seem to bother them. Today, it was melon seeds that were withdrawn. When it was the more usual sesame, I could imagine that there might have been bird droppings on the plants, but I can't see them getting inside a melon seed. Perhaps your team might enlighten us as to how salmonella gets into seeds and why nobody seems to get poisoned by it when it does. All the best, Steve.
Oh, boy.
I'm stumped.
Me too.
This goes back to this. It goes back to the listeria problem that we saw with melons before, but I don't know how it could get inside the melon seed. It may be associated with the melon seeds as a consequence of their being harvested, and I would assume that as the seeds are being harvested and washed, it's probably coming in with the processing of the seed.
Like if it was on the outside of the melon rind and then the knife brought it in and touched the seeds.
And that's effectively what we found with the peanut butter outbreaks,
hamburger
and all of these other things. It's really in the processing. In the case of whole animal butchering, it's in the processing where the intestinal contents mix with the meat, and then you have so many animals being put into the ground beef. And my suspicion is that's what's contaminating the melon seeds, unless they are truly internal to the seed itself, inside the cotyledon itself, surrounded by the seed coat. I would assume that would be a much more challenging experiment. And I'm not familiar with the melon seed issue that they've had in England.
Looking at an article from Purdue University, and they're talking about a variety, E. coli and salmonella within plants and seeds, and they say it's very hard to know because you have to cut the plants to look for the bacteria, whether they're, they get there from the cutting process or, right, just as we've said. So this is an issue. They may not, in fact, naturally be there. Who knows? Anyone know?
Please send a note.
Please send a note. One more from Varun, our friend in Bangalore. First, congratulations on reaching TWiM 100, and I wish the crew the same success that TWiV currently enjoys. I also want to vote up for Elio's latest idea of bringing in Talmudic questions as part of TWiM discussion. I want to punch in a couple of opinions. Microbiome, a product of big data science, has been a topic of great discussion. Recently, Eric Lander visited us and during the interaction had brought up this topic. Microbiome is a dynamic phenomenon, and variation between individuals is far more than what was actually predicted earlier. The understanding is that microbiome can change in the same individual depending on environmental factors. This discussion brought in two questions. One, do we know enough about the direction of cause and effect in microbiome context? Two, how to account for the variance in microbiome and its effect? In short, at least in a couple of cases, the microbiome effect has been shown to be the cause, but it is difficult to speculate how far this can be extrapolated. Second, there is a huge set of examples of microbes countering the strength of others, which is an important ability of residential flora. I projected that having a more diverse population will be self-controlling and more balanced. On the other hand, if the diversity is affected, allowing some strains to predominate, which in turn influences the normal process to a higher degree, tipping of the balance. This hypothesis allows variability of the microbiome to be accounted for. And he sends a link to a blog post in which he's written about this. In TWiM 100, the discussion of how a single mutation can make the difference was great. I felt you should have mentioned the Small Things Considered blog with the same theme, evolution of a super pathogen.
We should have.
You must remember that one, Elio, right?
Yeah,
that was not too long ago, this month, right?
It was,
it was in March.
Yeah.
I couldn't shorten the email further. I wanted to say it all. As always, you guys are great. Keep the science shows coming.
Well, I have to digest some of what he says because his point about the microbiome is very powerful. In order to give a reasoned response, give me a little time, though.
Well, the paper we just discussed on microevolution of USA 300 is really just looking at one microbe, and you saw how complex and how much work it entailed, and the microbiome is, in the case of the skin, is much simpler than it is in the gut. And so just imagine the logistical nightmare. You'd have to do the shotgun sequencing, and we really need to recruit a whole new team of both microbiologists and bioinformaticians to really do some deep think on how to address these issues. Maybe an FAQ, Michele.
We do have an FAQ on the human microbiome.
Oh, yeah.
So I encourage. It's a terrific one if you're interested. But he's right. He's hit a couple of the key themes that have emerged. That diversity in the gut microbiome is good, correlates with health, and out of balance dysbiosis is bad.
Mm-hmm.
But we clearly have a lot more to learn.
Which is good, right?
Yeah.
We like learning. And there's always something to learn. All right, that will do it for TWiM 101. And this episode and all the others you can find at iTunes or microbeworld.org/TWiM, or using any favorite podcatcher app for your Android or iPhone or tablet device. It's all there, and most of them are free. And do send us your questions and comments. We like getting them. TWiM@twiv.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
It's my pleasure. See you next time.
Thanks for contacting those authors as well. That's great.
Welcome.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
Again, my pleasure.
Always.
Enjoyed it.
And Michael Schmidt is at the the Medical University of South Carolina. Thank you, Michael.
Thank you.
Not just any university, but the medical university.
We've been here since 1824.
Wow.
Some days I feel like I've been here since 1824.
I'm Vincent Racaniello, and you can find me at virology.ws. I'd like to thank the American Society of Microbiology for their support of TWiM, and in particular Chris Condian and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.